Hi, my name is Jon Mai, and this summer I was extremely lucky to work with these members of the Smee lab looking at oyster drill predation on oysters. And so what I think is really cool about oysters is how important they are to the environment. They're ecosystem engineers. They provide structure and support for a bunch of different smaller benthic organisms, and they improve water quality, and they keep erosion to a minimum on the shorelines. However, what we've seen worldwide is that about 85% of our natural oyster reefs have been destroyed. A lot of this is due to climate change, pollution, but a main driver is predation, where up to 70 to 100% of a cohort could be lost due to predation. And so what the Smee lab focuses on is this idea of predator-induced defenses, where we can alter their physiology by adding environmental pressures such as predators, which will cue the oysters into putting more energy into their shell strength. As we see here, the size between an induced and non-induced oyster is much different. This is because the induced oyster takes energy into growth and puts it into shell building to have stronger shells. And these shells can be up to 50% stronger. And what we see in our past experiments is that these induced oysters have up to 300% higher survivalship against these predators. And this makes sense, right? The crabs can't crush the oysters if the oysters are a lot stronger. But it doesn't make sense for the oyster drills, which here in Mobile Bay is the main predator of the oysters. What oyster drills do is they put their proboscis near the lip of the oyster and inject enzymes and toxins that allow the oyster to relieve their clamp, and from there they will put their proboscis in and eat the oyster from the inside out, which I have here in these QR codes if you'd like to watch. But it doesn't make sense how that stronger shell actually improves survivorship against oyster drills. And so my goal this summer was to see whether or not there are other things going on with induction that is not described just by shell strength. And so my goal was to see how do oyster drills find their ideal prey, and how do induced defenses cause differences in predator choice. And to do this, I had two hypotheses. One being that if they're putting more energy into their shell strength, they're actually putting less energy into their soft tissue mass, which is supported by previous data. And with less soft tissue mass, there's less Q dispersal. So my first experiment was a choice assay where I used the Q dispersal in a current system where I had upstream oysters separated by a divider and let their Qs flow downstream to where an oyster drill would be. And from there, I tracked which side they went to and what was the treatment of the oysters on that side. The second experiment tackles the other hypothesis that if predator choice in prey is driven by prey profitability, or that there's a certain amount of reward for the amount of energy they have to put in, the drills are going to choose the oysters that have the highest reward relative to the handling time needed to eat them. And so with that, I have a feeding assay which I use I put basically a buffet of oysters for these oyster drills and saw which ones that they choose to eat, which ones did they attempt, and how many of these induced or non-induced oysters did they consume. And so with the flume experiment, we see that the drills visited the control oysters significantly more than they did the induced oysters. And when we looked at the soft tissue assays with those, we see that the controls had significantly higher soft tissue mass compared to the induced, aligning with our first hypothesis. The second experiment, what we see is that we have a bunch of different intensities of which predation behaviors are exhibited by these drills, where we could see them queuing in and using their siphon to discover them, physically visiting them and inspecting to make sure, it's not a pebble, it's an oyster. And then we see a predation attempt where they will use their proboscis or try to pull the oysters that I had glued onto the tiles off. And so what we see is that there is no significant difference between treatments, or induced or non-induced, with these oysters, but we do see a significant difference between the different classes of intensity, or the frequencies of those at least. This indicating that the oyster drills are displaying high, they're picky with what they eat. They're not just eating the first thing that they go to. And so when we look at handling success by the oyster drills, which would refer to them being able to pull these oysters off the glued tile and successfully eat them, we see that they're 55% successful with the control oysters versus about 30% successful with the induced oysters. And so with this last graph over here, we see that kind of the culmination of the whole story. Oyster drills choose their prey based on size. The control oysters, which are bigger because they have put more energy into that growth, are eaten at a larger magnitude than the induced oysters. And with that, we see that there is like a certain size range, which is about 26 to 28% of the oyster drill size of preference in oysters they could choose from. And so the results of this really go to show that oyster drills are conservative with their prey, and they use cues and handling to figure out which oysters they want to eat. And then we also see that the ideal oyster are these larger non-induced oysters where they have more soft tissue mass to eat with a higher success rate of eating. And so the next step in this study would be to quantify handling time and monitor these drills' prey for a longer period of time, so we could see like how much time is needed to break into these induced oysters versus the controls. And on top of that, measure abductor strength, going back to our original idea that there might be something else going on with induction aside from the shell strength itself. And so is this higher success rate in the controls due to a weaker abductor muscle? We don't know. And so why this study is important in the first place is because it shows that simple predators that we often overlook, like snails, have highly complex feeding behaviors. And that these induced oysters may be used in industry and for restoration projects because they show higher survivalship against predation. Thank you.